Types of staining. Staining in microbiology. What is staining? Staining is an auxiliary technique used in microscopy to enhance contrast in the microscopic image. Stains and dyes are frequently used in biology and medicine to highlight structure in biological tissues for viewing, often with the aid of different microscopes. Types of staining. Staining 3 तरह का होता है अभी. सिंपल स्टेनिंग, डिफरेंशियल स्टेनिंग, स्पेशल स्टेनिंग. सिंपल स्टेनिंग में कौन सा होता है? पॉजिटिव स्टेनिंग एंड नेगेटिव स्टेनिंग. डिफरेंशियल स्टेनिंग में ग्राम स्टेन एंड AFB स्टेन. स्पेशल स्टेनिंग में कैप्सूल स्टेनिंग, फ्लैजेला स्टेनिंग एंड एंडोस्पोर स्टेनिंग. सिंपल स्टेनिंग. Simple staining involves directly staining the bacterial cell with a positively charged dye in order to see bacterial detail. Example: some stains are commonly used for simple staining include crystal violet, safranin, and methylene blue. Simple stains can be used to determine a bacterial species morphology, that means cell shape, and arrangement, that means single, chains, cluster, etc. Negative staining. Negative staining employs the use of an acidic saint Positive staining. In simple positive staining, basic dyes such as methylene blue or crystal violet are used. The basic dyes gets attached to the negatively charged genetic material of the microbial cytoplasm. Example: positive staining technique has been used since the late 50s and the early 60s for enhancing the contrast of biological samples such as tissues and cell structures, viruses, etc. Differential staining. Differential staining is a procedure where more than one dye is used to differentiate between different types of organisms on a slide. This type of staining helps to differentiate between cell types and cell structures. Example: in a stained film, bacterial shape, arrangement, and presence of other cells like pus cells are noted. The two commonly used differential stains are gram stain and acid fast stain. Capsule staining. Number 1. Wet mount method. Using India ink, that is negative staining, the capsule is visualized as a refractile zone surrounding a cell. Number 2. Direct staining, dry mount method, that precipitates copper sulfate and leaves the capsule as a pale blue zone. Number 2. Flagella staining. These are stained negatively by phosphotungstic acid, that is PTA. Number 3. Spore staining. Number 1. By ordinary stain like gram stain, spore appear as an unstained refractile body within the bacterial cell. Number 2. By modified Ziel-Nielsen stain with 0.25 to 0.5% sulfuric acid as a decolorizing agent, spore appear as acid fast, that means red color.